Well, good morning then. My name is José Luis Ruiz, and I'm going to present you this tool that we developed at the Bioinformatics unit of the Institute of Parasitology and Biomedicine López Neira that is called reAnalyzerGSE. If you witnessed and listened to Edu's talk, it's somehow a development of his miArma software, but I'm going to let you know what we are aiming to do with that. First, just a little bit about myself. So I obtained my degree in basic and experimental biomedicine quite a few years ago now. Then I learned about omics data analysis and bioinformatics with a master's at the University of Vic and the Central University of Catalonia. And then I performed my PhD in the Institute of Parasitology and Biomedicine López Neira in the malaria epigenomics lab. Then I joined the Bioinformatics and Computational Biology unit at the Institute of Parasitology and Biomedicine López Neira that Edu already mentioned because he's the head of it. And then just a few months ago, I joined the genome informatics teams at the Functional Genomics Center in Zurich, in Switzerland. I'm going to talk about this work, this tool, this pipeline that we developed at the Bioinformatics unit of the Institute of Parasitology and Biomedicine. First, I just want to let you know what happened, why we tried to refine miArma and to create an easy-to-use bioinformatics pipeline. So as you know, there's supposed to be, or many consider that many researchers consider that there is a reproducibility crisis in science. This is just one example, but around 10 years ago, a huge survey by Nature, they reported that more than 70% of researchers had tried and failed to reproduce another scientist's experiment. And this is still a really hot topic. We can jump to a few weeks ago, actually, to last month, in 2025, for example, when a huge reproducibility project failed to validate dozens of biomedical studies. Or, for example, from early this year too, there was this Nature Communication editorial claiming reproducibility and transparency. What's going on and how can we help? Well, as you can imagine, bioinformatics and computing science in general can help, and they can help a lot. Bioinformatics may come to the rescue, but things are not yet there. For example, this is an issue by Science in 2011. They were already advocating that the way to go was to ensure reproducible research in computational science. And this was their main figure, which is showing us that full replication is possible, a gold standard in replication is possible in computing science or related fields, as long as the publication, of course, but all code and data, and also link and executable code and data are provided. Otherwise, reproducibility is not ensured at all. There's been many examples of different recommendations, for example, by PLOS Computational Biology, 2013, 10 Simple Rules for Reproducible Computational Research. Also more recently, in 2020, there was still a need for recommendations for improving reproducibility in computational biology research. There was even this challenge in 2020, like, okay, you may think that you did it properly, but they created the 10 years reproducibility challenge. Like, hey, your code from 10 years ago is still working. And I'm not going to go into detail, but spoilers, mostly the code that we did a decade ago is not working anymore, mostly. There are still plenty. This is still, again, a hot topic. There's plenty of recommendations. I'm going to focus on these guidelines published in Briefings in Bioinformatics a couple of years ago, because they are clearly stating that there are 5 pillars for achieving reproducible computational research or bioinformatics pipelines. So there has to be, of course, a continuous validation, but you have to ensure that you are getting literate programming, code version control and persistent sharing, computing environmental control, persistent data sharing, and very important, the documentation. I'm sure that there are many bioinformaticians in the audience today, and you know what I mean. Sometimes documentation is very poor, or some of the bioinformatics pipelines or software that are shared are lacking some of these pillars, and then the situation is much more difficult. This is also important and is something that we incorporated in our latest pipelines, including the new version of myArma and reAnalyzerGSE. The fact that you can use containers, you can enclose your applications in a container, and this has many, many advantages. For starters, you are building onto a hardware, of course, and a host operating system, but then you get a single container engine, and you can get really reproducible apps that are just contained into that closed environment that you have created. This makes tools really shareable and your research really reproducible. Beyond reproducibility, I want to talk briefly about the FAIR guiding principles. You may know about these. They have been around for around 10 years now, but the point is, FAIR, this is coming from F of findable, A of accessible, I interoperable, and R reusable, that all data in general, but in particular in research, it has to conform to these principles. So it has to be findable, accessible, interoperable, and reusable. I want to, all of them are really important points and very interesting, but I want to focus on the reusable one, because your research should be reproducible, of course, but the data that you generate not only has to be, any other research should be able to get the same data and obtain the same results, therefore reproducing your results, but that data should be also reused for many different purposes. Now, reaching to transcriptomics and building onto myArma again, we felt that there was an opportunity there. For example, you may know Gene Expression Omnibus. This is the NCBI most famous database, most popular database to storing omics data. So it has been also around for a few decades. The latest update paper, it was from last year, after 23 years. And the point is that there are now, well, this is from that paper 2023, so now 2025 should be more, right? So there are around 25,000 RNA-seq studies storage there, publicly available. Luckily, now journals are frequently requiring, and it's mandatory, that the sequencing data is made public in repositories like this. So this is publicly available. They are aware that people are downloading that data beyond the original hypothesis. So this reusability concept, it means that you can perform a reanalysis, but not looking forward to achieve the same results, but in order to implement and to analyze roughly new hypotheses of any kind. This is part of the GEO paper, as they stated clearly, people are analyzing data in ways not anticipated by the original data generators, and there are many examples of this reuse of the data. They have tried to help users to do that. For example, they have created GEO2R in 2020, and from a Gene Expression Omnibus entry and a sequencing dataset, you can already obtain easy to interpret and ready to use analysis, plots. But the thing is that these are still steps in the good direction, but these are still lacking reproducibility. So the situation in transcriptomics can be summarized as a situation where there is a lack of standardized, fully reproducible and easy to use transcriptomics pipeline. There's a lack of comprehensive methods reporting. For example, it's difficult, this GEO2R, to get an idea of the fine details and the fine parameters that have been used, and which of you, of course, as you know, may greatly impact the results. And at the end, these sequencing data are often transcriptome or genome or transcriptome-wide assays that are not being fully investigated, right? They are just leveraged for a single hypothesis linked to a particular publication, then they are placed on the repository, and there's that. And they're not going to be used again. So it's important to try and to leverage all of the already existing sequencing data that is there just waiting for researchers to think about it. We developed then the reAnalyzerGSE pipeline, as the name is based on this concept of reanalyzing directly GSE entries. These are the geo sequencing datasets. And this is already published made publicly available on bioRxiv a few years ago. We have kept updating this and building into this, of course. There's a GitHub repo that we are trying to keep updated. This would be the QR code. And I just want to show you that, again, this is actively maintained. Latest version is already, after 52 releases over the years, is already version 3.1. And even if we are still lacking, I mean, this is a work in progress and we are still lacking a proper wiki, for example, even if we are still lacking this detail how to do, there's still plenty of information in the GitHub, plenty of examples and test data, as I'm going to show later. So you can already try to install the pipeline and see what's what. This is the general overview of the pipeline. Again, it's still being updated. New tools are incorporated. This is a bit outdated, actually, but it can serve me to briefly explain you about the different modules. So first, as you may be also aware, if you have ever performed a bioinformatics analysis, the data loading, handling the data, converting between different formats, preparing the data structure, the folders, everything, that can become a nightmare. So the first step was to make sure that in order for the pipeline to be as user-friendly as possible, we made the data loading as easy as possible. So this is very easy. You just have, you only have to get a public repository ID, or if you already have local raw data, the FASTQ sequencing files, and of course some metadata, which conditions are these files, whatever, then you can already execute reAnalyzerGSE, provided also, as Edu mentioned, that your organism, it has some reference sequences available. So of course, the alignment step in the typical RNA-seq study pipeline, as Edu mentioned, can be performed. After data loading, there's a step that is also very important, that is data preprocessing. So data may have to go through decontamination. It may have to go through ribosomal RNA removal. You know that it depends on sequencing, but ribosomal RNA may account for the most part of your sequencing data. And you may also need to downsample and in order to normalize your data, because maybe one condition was sequenced way more or in way more coverage and depth than other one. So that may impact results down the line. And then the data processing step, which is of course the core of the pipeline. You can go, if you have microarrays, and these tend to be really platform specific, right? Or they may depend on the particular microarray used, but we did include when we were requested to the different microarrays. So all the canonical typical microarray processing steps are performed. And if we go through the RNA-seq pipeline, I'm not going to go into detail because Edu already covered this, and miArma, an updated version of miArma, is taking into account all of this coming from messenger RNA-seq data. One thing that is a work in progress is to fully leverage the capabilities of miArma, which is very powerful, as Edu mentioned, to analyze all the microRNAs. But for now, only messenger RNAs, transcript-based RNA-seq studies are supported. So as Edu mentioned, I'm going to just talk about one of the improvements that we can apply into reAnalyzerGSE and miArma, which has to do with the filtering. So of course, you may know that a typical density plot of the raw counts after an RNA-seq study looks like this. So this means that we're briefly in the y-axis, of course, we have the density, and in the x-axis, we have the expression level. So for any given transcriptome, this is the truth. The great majority of the genes are not expressed. Okay, so the distribution is considered to be binomial, but it's actually cut here, but there's a huge peak because near close to 0, or this was already in log scale and normally, so we have this negative value here, but the point is the immense majority of the genes are not expressed or are expressed at really low levels. And then one of the first steps when you're analyzing RNA-seq data is to filter this, to get this out, because you are considering it noise. If you have only few hits that are aligned to a gene that is really poorly expressed or is not expressed at all, but you may still get that unspecific alignment, then that's considered noise and you're supposed to discard that. But then the question is, where do we cut? Here? Maybe. Why not? We could also cut here. Maybe. Why not? We could also cut here. Maybe. Why not? Right? So this is a hot topic, actually, because if you remove, you're going to remove everything below a threshold. So the genes that could have got some of those sequencing reads would appear as non-expressed, but maybe they were expressed at really low levels, but you just cannot detect it because you are at the edge of the sensibility and the detection threshold of RNA-seq studies. So this happened, again, this was developed in the context of the core unit at the Lopez Neira Institute. So we realized that some researchers were actually finding this. For example, they would perform knockouts studies of animals, knocking out very important genes, for example, neuropeptides, for example, hormones. But if those genes were, and of course you would get the phenotypic effect of that knockout and whole research lines. But then if you perform knockout versus wild type RNA-seq studies, you wouldn't detect the gene that was originally knocked out. So why was that happening? It was happening because of this filtering, because we in the field, we tend to assume that lowly expressed genes are not important and are not impacting phenotypes, but that's not necessarily true. So with reAnalyzerGSE, we implemented within miArma, the updated version of miArma, this being filter. And here we are just showing for the reanalysis of a geo entry how this gene, which is encoding for a neuropeptide called cortistatin, which is really relevant, but is expressed at really low levels. So applying the filter that is more common in the field, the standard filter, this gene was discarded as noise, so the value would have been non-assigned. Actually, the gene wouldn't have been even in the tables. So this is a small, subtle difference, but it's not the same that a gene is not in the table or a gene is not expressed, right? But people without bioinformatics expertise or knowledge, if they didn't find their gene at the table, they would think, okay, it's not expressed. Well, maybe it was expressed, but at very low levels, and it couldn't be detected. With this filtering and disregarding, it doesn't matter the aligner that is used, we could restore, we could recover the detection of a meaningful expression level of this neuropeptide. Finally, after the data processing step, typically what you would ask of a transcriptomic analysis is a differential gene expression design, right? So of course, differential gene expression, functional enrichment analysis, also network analysis, all kind of plots that may be of interest, finding biomarkers, housekeeping genes, all of these can be done easily with these latest steps that is working on the automatic output of the miArma module and the data processing step. I'm not going to go into detail in this, but if you're interested, we can discuss, or we are always happy to be in touch or to answer GitHub issues and to keep the software updated. Regarding the usage, As I mentioned, we went for a containerization, for a container solution, which is called Aptainer. This is the latest update of what used to be called Singularity. And it allows you to create a self-contained operating system, and then with a one-liner, you can just invoke the software that is already prepared within the image, and you don't have to worry about installing dependencies and basically anything beyond installing Aptainer, which is already available in most high-performance computing and HPC clusters and bioinformatics desktop machines. So this is a snapshot of the GitHub. All the manual is there. The help page of the software got quite a bit complicated, even if we try to be verbose and to be comprehensive in the explanations. Of course, it can get really complicated. So we also provided some quick start and minimal examples, okay, in order to analyze both local data or to reanalyze geo entry. All the instructions are there. But, and as I mentioned, you get just a one-liner. But of course, it would be crazy to decide which options do you have to choose and to manually enter them. So it would be a one-liner command line, but then it could be really long, right? So similar to the configuration file of miArma, there's a configuration file in reAnalyzerGSE where you would already get default values for most of the parameters, and you can just go ahead and input your sequencing reads, your organism, here in reference genomes, whether you have already, for example, created an index file for the genome, whether you are interested on a particular gene, so you would already get plenty of analysis regarding that gene and figures and plots and everything. So at the end, you get a really well-structured, or it's always the same, a really well-structured results folder, okay? For example, within the final results reanalysis is where you would find the output of the differential gene expression analysis, all possible comparisons. Of course, you can fine-tune this, but the default situation is that all possible comparisons are performed, and you would get the corresponding, for example, volcano plot for each of them, the corresponding functional enrichment analysis, housekeeping genes, the differential gene expressed genes, of course, the DEGs. Also, you get quality control, all of your transcriptome quantification, right? Normalized with different approaches, violin plots. So we try to be comprehensive, and everything should be ready to use at the end. We also structure our results following creating a final report. So you would get a web page following roughly this format. So you would get a click-through guidelines to interpret your own results, summary of starting with a summary of the samples and the experimental conditions that were analyzed, summary of the comparisons and covariables that were used, if any. Here, for example, in the test data, no covariables or potential batch effects were included, of course. The layout and strandedness, this can be actually automatically predicted, so you don't have to worry about the kit that was originally used in the library preparation step, whether the strandedness was reverse or forward or whatever. Then you get your quantification, the counts here, these huge tables, right? Also violin plots, and you get a list of differential expressed genes for different comparisons. Here in the test data, again, just a generic condition A versus generic condition B. You get the number of differential expressed genes, this ahead at the top 10, ahead of this, just sorted based on the difference, top 10 in each sense. You can, of course, download all the information, all volcano plots, all functional enrichment analysis, housekeeping genes, and quality control analysis. These are actually very important because these are very different files that frequently, based on our experience on the bioinformatics unit, we would need to perform a first run of reAnalyzerGSE and then double check the quality control and the results of these tables, and maybe change a few parameters, and then rerun reAnalyzerGSE again, which is actually the main use that we have for this pipeline, just to let us do quickly what the users would expect to be done really quickly, right? So of course you get a multi-QC report. As you know, multi-QC is not only accounting for the fast QC and the raw data reports, but it can get everything, information about the alignment, getting into account QualiMap, deep tools, SAM tools, the aligner, the aligner software itself, for example, here Star. You get everything in an aggregated way that is really useful to have. You also get this QualiMap report with the multi-BAM QC mode. So in this case, for these 4 samples, you are already going to get all of the typical QualiMap report that is telling you how the alignment performed. And we created some PDF files ready to use with multiple different quality control figures. I'm just going to show you some of them. This is the first one that is included because, as I mentioned, it may depend where you did you cut, and then the here, for example, as these were just test data, this distribution does not look good, right, after the filtering, but it serves indeed as an example about how a filtering can greatly affect the distribution of your count data. Okay, you get an assessment of how the normalization worked. Here, of course, with these mock data sets, the normalization did not work at all, right? And this wouldn't be acceptable in a typical example. You get information about the library size. You get also a replicability analysis. So we may have told the software, hey, I have 4 samples and 2 for each category. But then if there's, for example, if something went wrong on any steps of the wet lab analysis experiments, or maybe if there's a huge biological variability, then if you don't get a difference between the 2 conditions, you cannot perform differential expression analysis, right? So first, to assess that, we can check out the correlation between the 4 samples in this case. We can also check out how a principal coordinate analysis, the multidimensional scale approach for reducing complexity, how it's showing us that the samples clustered. Here we see 1 and 2 in one condition, and 3 and 4 clearly differentiate, right? We are also showing a PCA, principal component analysis, alternatively, that is showing us the same. We are also showing how the most differentially expressed entities are behaving. This can give you many clues about what's happening. And finally, we are also showing unsupervised hierarchical clustering results with these dendrograms that can also, of course, be automatically grouped. And these now may seem silly because these are just 2 conditions and 4 samples. But when we have requests on the core service about hundreds of samples, then it's really useful to have this kind of clusterized samples, so you can double check whether the conditions are behaving as they should, or there's a mess, right? So anyway, that could be the brief overview that I wanted to show you about reAnalyzerGSE. Again, this is a work in progress. We are happy to keep supporting this pipeline. Any feedback is welcome through any channel. The GitHub issues are away, or our contact data is easily can be easily found also online. Just to let you know that our to-do list or work in progress is including to fully extend beyond mRNA-based assays. For example, miRNA is very powerful to do that, or circular RNAs, isoforms, splicing, whatever. We can also support more statistical tests and models. We have been trying to do that when there are advances published, like new statistical methods, new recommendations, new guidelines. We are trying always to incorporate them because this is being used in the everyday work of the core service. We have to create a proper wiki and detailed documentation on the GitHub. Although if you have already bioinformatics experience, it should be easy to follow already. We can improve the parallelization and do some other performance tweaking. And we would also like to get it published in an index journal soon. And yeah, we are also happy to show this on conferences and so we can get core services to use it, as they are also using Miarma, and we can support them, both core service, but also final end users, no matter the bioinformatics expertise that they may have. So in our center, for example, we have been also supporting students and people who, instead of asking us for the analysis, thanks to this pipeline, they are already capable of doing the analysis by themselves and getting very useful insights on their own data or the data published by others, for example, regarding diseases, clinical samples, or whatever. That was all. Thank you very much for your attention, and I would be happy to try and answer any question that you may have.